welcome more times. We've got today is fantastic. The whole festival, Patrick's Festival as we call it, has been fantastic, and Coffee Buddies has been double fantastic. So what more can I say? I should go now, but we've got a great lineup today. Today is something special. It's called the XY Factor. It's a chance for startups, scale ups, any ups to come and present what they've got, but through the interview media, which is me asking them questions. And then we've got a group of people here who's going to judge the competition. Graham, can you just say who the people are and introduce our panel?
Yes, this is sponsored by MSD, and it's wonderful to have our regular community member, Michelle DeBar, here. So do you want to just say hi to everybody, Michelle?
Yeah, sure. Hi, everybody.
Great stuff.
Fantastic.
And we also have Michael Salako from Start Codon. If you'd like to say hi to everybody, good to see you.
Good to see you.
And we got the legend that is Tom Blackburn.
Tom Blackburn, yeah.
Who's also going to be one of our judges. Thanks, Tom, for coming.
Hi all.
Great.
So lovely to see you, and it's lovely to have a mixture of people. Remember, Coffee Buddies is all about entertaining, informing and engaging, and so is the XY Factor. So you never know, it's a cameras on event. You never know when I might say your name and we'll bring you in, but we bring you in with aplomb, so you won't be sort of dumped in. And today is a fast. We need to be fast in how we operate, because today we've got seven super
companies, yeah,
that have come to see us. And so our goal is to move through the companies. We're going to give about 10 minutes to each company in an interview process. Then we're going to give the judging panel a chance to ask a couple of questions, and then we're on to the next one. And then at the end of the event, it will be the end, and that's fantastic, isn't it? There always has to be an end. So what we're going to do is we're going to kick off straight away.
But we're also going to have two winners at the end from Tuesday and Thursday. And then we're going to be presenting them on a Friday night at 6 o'clock UK time. So there will be a first and second place, and that's what the judges are marking.
And this moves on to the bigger thing, which is called the Best is Best. And Graham, do you want to explain that?
Well, we're going to continue this maybe on a 2-monthly basis, and also at the Biotechgate Digital Festival in September and November. And then in December, we're going to have a final, the best of the best. So we hope you make it all the way there, everybody.
So keep doing it. If you don't want to go to Strictly Come Dancing, this is the second best. So here we go. We're going to start off, and I'm going to introduce, Graham's going to correct me if I get the names of the people wrong, because he always corrects me. But we've got Shahar first. So I'd want to welcome Shahar as our first person up to talk today. Shahar, can I welcome you and say, who are you, where are you, and what do you do?
So I'm Shahar Keenan. I'm a CEO, a co-founder of Polaris Quantum Biotech. We're located in Raleigh, in Durham, which is a suburb of Raleigh in North Carolina, which is the east coast of the United States.
Fantastic. It's so lovely to see you. And we've met before, which is perfect. But maybe you could just tell us what your company's about.
Sure. So Polaris Quantum Biotech, what we're doing is we are using quantum computers, AI, and machine learning to develop drug leads at scale. So we develop them on the computer, and because we are using quantum computing, we can look at very large chemical space. So for a specific biological target, we build a bespoke, a chemical space, very large, 30 billion molecules, and we find the right molecules in it. And these are molecules that can become good drugs. So we're not just looking at would it bind to that specific protein, but we're also looking at drugability. And if you're interested in specific properties of that molecule, for example, should that molecule pass the blood-brain barrier or never pass the blood-brain barrier? What's the level of toxicity and these kind of things? Yes.
Very simply then, you've got data. You've got, if you like, a framework for deciding what are the properties, and then you've got this enormous computer. Is that the idea?
So the computer is not, well, it is very big, but the time it takes to run is very fast. Because we're using a quantum computer, we can search, and this is an optimization problem. It's not even a search problem. We can optimize a molecule looking at 30 billion, a library of 30 billion molecules and find the best ones in it. And it takes us, the run itself is very fast. It's a 1-2 minutes run.
Fantastic. And you're a company, you're incorporated. How long have you been incorporated?
So we are a year and 2 months. So we've been, a year and 2 months. So we still think of ourselves, of course, as a young company. However, we've done a lot with that time. We've raised some funds. We have a team of 5 people with several consultants and have customers as well that we work with.
If you looked in front of you into the next 3, 6, 9 months, where are the pieces of value data that are going to come, and what are they going to tell? So if you've got an investor, what are they going to see that's coming that gives them the sense of momentum?
So we are building 2 things. We're building our team, but the main thing that we're building is the automation of our platform. Because what we want to do is to do this at scale. We want to look at multiple targets, develop multiple assets. And the way to do that is to build the automation of our systems. And that's what we're doing now. So in about 6 months, we want to have at least 2 assets that we developed that have finished synthesis and measurements, and that we can start publishing what we've done, maybe write some of the IP on those and start licensing those to customers.
So when will you, so really the validation is when your customers buy, and are you getting feedback from the customers now?
Yes, so we already have a customer, and that customer has signed 3 projects with us. We're finishing now the first project. So although we are doing a lot of work with customers, we're also developing our own internal assets. We're doing both of these things. And we are looking for other customers, mainly small biotechs.
Yeah. And how much money are you looking for to get you through to the whatever you call the validation points or the proof of concept? What are you looking for?
So right now we're raising $3 million. And that should get us for the next 18 months. However, we will have a validation way before that. Probably in less than a year, we will already have packages that we can sell or license to pharma.
Yeah. And if I said to you, what's your secret sauce, Shahar, what would you say the secret sauce is?
So our secret sauce is we know how to look at a protein binding pocket and build a large virtual chemical space that is relevant for that pocket, and then search it. So it's not enough to build the library of molecules. It's also important to find the right molecules in that. That means that we find molecules that are very difficult to find from large virtual chemical space. And this is the sauce we have, yes.
And do your customers give you a specific brief, like they want you to find, I don't know, ubiquitin, they want something in the ubiquitin pathway that could be used in oncology? Do they ask it that sort of, is that the question they use?
So what they say is usually, this is the protein, we've tried other methods and they didn't work, help us. But what they're looking for is a set of small molecules that bind to this protein and disrupt the activity of that protein. But these molecules need to have some specific properties. For example, if they're looking for a small molecule that will replace a biological drug, okay, so they're looking for molecules that will bind in that pocket and disrupt protein-protein interaction.
Where's the data set that you, if you like, look at? Where is that? Is that a public data set or?
So we are building our own libraries. We are using quite a lot of information that is available, like chemistry kind of information, what kind of groups bind to what kind of scaffolds are available. But based on that, we are building our own large libraries. We're building them and we're collecting them. So one more sentence, in about a year from now, we will have one of the largest data sets of molecules in the industry, including what kind of protein these molecules bind to in a high accuracy calculation. So in a year from now, our data set is going to be something that will be relevant for the rest of the industry.
Fantastic. So Shahar, fantastic. And I don't want to cut you short because it's such,
no, please.
I could talk to you all day about this, and because it
could answer.
It
yes.
sounds like you're on the edge of something really great. So can I just ask our panel of judges, got a couple of questions, maybe you could throw the questions in, please. Graham, you're on mute.
Tom, yes, if you'd like to ask first, Tom.
Yeah, it's a fascinating approach, and I've heard so many other companies, you know, with AI, but is the differentiation between you and them chemical companies just the quantum computer? Because other companies are using quantum computers too.
So the problem with AI is that you need a really large data set. That kind of data sets are very difficult to find in chemistry. You'll need to have billions of molecules in order to parameterize your AI to have an accuracy. You can do it for a small set, but then you cannot do it at scale. Every time you work with a data set, you have to tweak it. What we're doing, because we are looking at such large chemical space, and because we're not using AI, we can do the same procedure for whatever protein we work with, and we can do this at much larger scale. Now we are using quantum computers right now. The quantum computers that we are using are not the regular quantum computers. So these are not gate computers, these are annealers. They are available and other companies can run them. The algorithms that we use are unique to us.
Fantastic. One more question. Michelle? Or
Michelle, yes, Michelle, and then Michael.
Michael?
Yeah, Tom actually stole my question, so thanks for that, Tom. I'm also thinking more like your business model. I understand you're thinking of a sort of dual business model, like your own pipeline, your customers' pipeline. I know from investors' perspective, they're typically not very keen on dual business models because you need to focus your attention on one or the other. What would it be that you would select if they decide you have to go this direction?
So it's very easy. If somebody will pay me enough, I will not work with customers. I will just develop my own pipeline, but that's expensive. So that's why we're building those things. In 2 years from now, the part of, or the percentage that we will do as a services is going to be very small, maybe not existent. But right now, for the next year or so, I do see us looking at both business models.
Fantastic.
Right, Michael, you've got a question?
Yeah, no, definitely, sure. Thanks so much. Really exciting. I've seen a company recently, Racco, that takes a quantum computing and the AI approach. And I've been looking at this area for a bit of time now. Are you actually starting from? Do you need information on the sort of protein structure, and then you're looking at in silico what molecules can fit into that pocket, and using the quantum computing, you can, work out, the probabilities of that. And then with the AI, it helps on that training set, because, I've always wondered whether if you could have a big enough computer, could you fire just, known molecules and the like at a pocket, and, due to binding energies and the like and the feedback you get and the modeling, you should be able to track back from that to work out roughly what molecules should fit in that pocket. So I'm just trying to understand how your platform approaches that.
Yes. So right now we are limited. Even quantum computers, they are not that big. They have, a couple of thousands of qubits. So that's why we can only work with 30 billion molecules. The bigger the computers, when we reach 10,000, maybe 20, 30,000 qubits, then we can really look at very large chemical spaces, maybe even the whole of chemical space. That's where we want to be. Your description of what we do, finding the binding pocket, finding molecules that fit in that's where we are. Yes, indeed. So far, we're building our own libraries just because we are limited by the size. Even 30 billion molecules is something that we think of as limiting. We want to get to the whole of chemical space.
Fantastic.
Right.
Graham,
well, thanks very much. That's
Shahar, a real pleasure.
Pitch. Thank you very much.
It's a delight to talk to you as always, and we're moving on, and hopefully you'll keep us updated and come back. Shah, because we really love it. The next one up is
Ishay Attar, yes.
Now, Ishay, where are you? Because I'm trying to find you. Where's Ishay?
He is on the screen. He's there.
Fantastic. So,
Can you hear me?
I got you. Fantastic. So, welcome, Ishay. A big welcome, a big coffee buddies welcome to Ishay. Ishay, where are you? Who are you? And what do you do?
Well, first of all, let me ask if the sound quality is okay. I'm a little bit. I'm sick,
very nice. It's
so
perfect.
my voice is not at its best. You can hear me? Okay, great.
Yes,
perfect.
yes. You're not contagious, are you?
Okay.
No, I was joking. Okay.
Yeah. Okay, Ishay.
Yeah. So yes. So my name is Ishai. I'm a biomedical engineer. I'm from Israel. I'm a serial entrepreneur. I founded several companies. One of them was acquired by Becton Dickinson, Bard. My main specialty is biomaterials, and that's what I've been doing most of my time since 2007. And this time, I decided to solve the problem of tissue scaffolding, tissue engineering, and regenerative medicine. And this is what we're doing in BioChange since 2017.
Okay, perfect.
This is perfect for you, Tony, actually.
What's that?
You know, the scaffolding and the engineering the face.
Anyway, back to Ishay. I can't believe you'd say that. Anyway, Ishay, back to serious matters. So you've got a company. How long is it incorporated? Have you had seed funding? Where is it? Is it at the beginning or the middle or the end?
So we are about to start the clinical phase. We're preparing for the clinical phase of our first product in second quarter of 2022 next year. So we already have achieved the preclinical, very impressive preclinical results that what our scaffold can do, which is very unique, is that it stimulates the tissue to repair itself. So as you know, we humans, we don't have the ability to regenerate tissues all that well. So we age, and some of the tissues break apart. Some of them are destroyed because of illnesses or cancer or other traumas. And there is no way to regenerate the tissues. Well, there is, but it's very, very limited currently. And we have a scaffold that's an injectable formulation made out of a protein, cross-linked protein that it's cross-linked into a foam. And once this is injected into the body, various organs, it stimulates the tissue to regrow itself. It disappears within about 3 months.
What's the stimulation? What stimulates what? Okay,
so the scaffold is made of cross-linked gelatin. So we take this known biomaterial that's been used in the medical industry since the 1950s. It's very safe. But the new thing is that we are cross-linking it with some enzyme into a foam structure. And this foam structure is exposing all those integrin attachment sites, the cellular attachment sites of the gelatin. And that really attracts the cells from the tissue, that they come in from the periphery they grow into the implant, into the scaffold, and then degraded and replace it. Everything is synced, the buildup of the new tissue and the degradation.
Is it scar tissue, or is it, so what sort of tissue is it that is it a granuloma? Is it a granuloma?
No, great. Excuse me, great question, because we actually did a lot of verification studies, immunohistochemical evaluations of what kind of collagen, what kind of tissue is being replacing the scaffold. And we discovered that the tissue that's replacing it it's a healthy tissue. It's like the young version of that person, of that animal, because we're still in animals, okay? So we see collagen type 1, we see elastin fibers. I'm talking now about skin indication. And we see it grow. The remodeling phase takes about 3 months, and then it stabilizes, and there's new tissue there, bulking or replacing becoming a new skin, and it doesn't change anymore. It doesn't grow after that phase.
So there's no inflammation.
There is always inflammation. Inflammation is part of the story. When you inject a foreign body into an organism, you get an inflammation. The question is, what type of inflammation? Is it destructive or constructive? What happens? Is it, and that we have we've fine-tuned this to have the right type of inflammation that builds the tissue, that supports it, that grows it, and doesn't do any. And it's very mild inflammation. Yes.
Okay, fantastic. So, and your inventive step is the. I heard the word enzyme integrin. Is that your inventive step?
Yeah, the inventive step, and by the way, we just were allowed the patent in Europe yesterday, and we have one in the US granted. So the inventive step here is using the gelatin as a foam, cross-linking it into this porous scaffold structure that allows all those cells to work with it.
Fantastic. And how much money how much have you raised already, and how much are you looking to raise?
So we're a strange kind of company, not standard biotech. We raised only $600,000 US so far, okay? And we had a strategic deal that placed several millions of dollars in the company. And we have sales in veterinary, okay? So we sell veterinary products right now for urology. In veterinary, we injected the urethra and we solved the problem with urinary incontinence, which is also gonna go to humans later. And that's one product we have. Another product that we are bringing veterinary to the market is for periodontal tissue, for to repair the gum tissue, regenerate it. And we also have a product that started now to sell we have orders on a product that we sell now for stem cell delivery. So we also are active, and we also want to expand this to bioinks and stem cell delivery. You know, if somebody wants to deliver his stem cells into the tissue and have them survive longer and give them a place to survive or better chance, then we would provide it with our technology, with this injectable scaffold.
So you've got a multi-layered business of which you're stimulating, I don't know, injectable scaffold is one component. So how much are you trying to raise?
All right. So right now we're raising $10 million, okay? So far we were at sort of a bootstrapping mode, and we did deals and it worked. Right now we want to take the first product to the clinical phase, which is a product for aesthetic dermatology and plastic surgery. That means that we will be able to inject this to aged people and regenerate their skin and make them a young skin again. And this, yeah, and that's like sounds science fiction, but we have very strong preclinical evidence that this can actually be a game changer in aesthetic dermatology and plastic surgery. Plastic surgery, if there's a tumor removed from the chest of a lady, then she would want to recover this.
I must ask you, will it be new skin or young skin?
New young skin. You know, we're gonna make the world young again.
The guys in apoptosis may have another opinion. So let me just ask you. So to an exit, are you quite? Would it be a short exit? I mean, you sound like a big multi-layered business. Are you exiting? How are you going to exit?
So, so we're a platform company, like various other ladies and gentlemen, startup company that presented here. The platform is we're growing it. We have several collaborations on various indications in early stage. And the first one's gonna go up is in the aesthetic dermatology and plastic surgery. And there we have a line of strategics interested to take this product to the next phase. So we need $10 million to get it through to big deals that would allow us to grow the company from those incomes from the milestone and upfront payments from the aesthetic dermatology product.
Fantastic, my friend. Well, we wish you all the best. We thank you very much. I'm going to ask for a couple of questions, Graham. Maybe you can curate the questions from our panel.
Absolutely. We're gonna start with the person that needs it least and then work our way up. So work out the order. Michael, you go first.
No, thanks so much, Isha. Just a general question in terms of IP situation here. So you mentioned that you filed, but did you is this your own technology that BioChange have generated, or did you in-license any background IP?
I'm the inventor and the CEO of the company. I invented everything myself.
And was this done at an institute, or you just literally?
No, like I said, I'm a serial entrepreneur. I already did one exit. I used the capital to fund my own early stage development. I bootstrapped the company, invested my own money. That's why it's a different story than usual. Okay, and I also know this space very well. I mean, I've been developing products from gelatin since 2007. So I can modestly say that I'm one of the persons that knows best gelatin in the world.
So the ECM side, you've got background in that, from sort of lab background, scientific background?
Both. I'm a business and scientist.
So in terms of the sort of licensing deals or money to revenue that you could bring in off of this, invest 10 million investments. Would it be? It's a platform, you say, but and you mentioned incontinence and other areas, but assume it's underpinned on that. I'm just trying to get an idea of what you would license to different companies. Would different companies ask for something particular, and then you can use your 3D foam sort of platform to go into that or address a need, or you have off-the-shelf products that you could just license?
In veterinary pet care, we have off-the-shelf products that we are licensing. We haven't done that yet in pet care, but we could. But mainly, I think if we look at the main product that we're going forward with this investment, the aesthetic dermatology, we already have strategics, large ones, interested to license or make a commercial deal that would allow them that the product is exclusivity in certain territories, like US exclusivity would be worth around an upfront of $10 million, and then a milestone payment. And we're in negotiations around that now with various global players and local players, and it's interesting.
Right. Okay. Well, I better go to Michel next, otherwise I'm going to lose him as a sponsor. So, Michel, would you like to go next?
Thanks, Graham. And it's clear where you placed Tom and myself. But so, thank you. Say that was a very interesting story, and I think there's definitely different markets for it. And coming to those different markets, if you think of aesthetic dermatology, which has a very different regulatory track and reimbursement possibilities than, for example, where you look for plastic surgery from patients that have undergone, for example, surgery for tumor removal, where you have clearly a different need and also a different regulatory framework, but also reimbursement and pricing. So I don't see those as overlapping markets, but if I listen carefully to you, you look at geographic deals to companies that cover everything like that. But can you maybe touch a little bit more on that? I think it also links into what Michael meant, like, are you looking at carving out certain areas in your licenses to different groups or companies? I just don't comprehend really your commercialization strategy.
Exactly, carving out. Yes, exactly that. So if we're, usually the plastic surgery and aesthetic dermatology, they come together. We cannot carve out this one from the other, but we can carve out the urology product from the aesthetic dermatology and plastic surgery product, even though it's the same formulation. Okay, it's very similar, but it's going to be branded totally different physicians, call points, everything. So and different strategics usually, okay, that would sell this versus all that. The greatest thing about, and that's why we went to aesthetic dermatology first, is that there's no reimbursements, it's out of pocket, it's a booming market with double-digit growth rates. It's amazing market to be in.
Fantastic.
Right.
I think we have to move on, but I think
we'll just Tom, just a quickie.
I want to look like Mark, by the way. What questions do you find most difficult from the VC, when you're presenting your case?
What is your valuation, and why do you want $40 million pre-money if you only raise 600,000?
Right. And what is the answer?
The answer, look, I tell them, don't look at what I've done. Look at what I've achieved. I've achieved not what I've raised, but what I've achieved. I've achieved with 600 this much, and I got to a point that other companies benchmark are worth around 100. then I shouldn't be penalized for it. I should be, you know,
celebrated.
Rewarded, celebrated, yeah.
So this regeneration of tissue then, is this the N-glycans associated with gelatin?
The aminoglycans, you mean?
Yeah, N-glycans and gelatin.
Yes, gelatin has an abundance of RGB motifs on the molecule. On you know, it's derived from collagen. It's a variant of collagen, but it has less immunogenic response. You know, gelatin doesn't exist in nature, but it has all the good stuff from the collagen without the bad stuff from the collagen. The only problem with gelatin was that, as an implant, was that it was difficult to work with as an injectable because it's solid at 37 degrees. And below it, it's a solid hydrogel, so people didn't work with it so much. And I know how to work with gelatin.
Fantastic, Graham. We need to.
Thanks, Ishay. Really appreciate that.
We appreciate you coming. Lovely to see you, and we'll see you next time. We hope. And moving on, because
we got Sergey. Yes.
Sergey.
Hi, Sergey.
Where are you, my friend?
From Crystals First. Nice to see you.
Lovely to see you. So let's just hear. Who are you, Sergey? Where are you? What do you do?
Hi, my name is Sergey Glinka. I'm co-founder CEO of the company called Crystals First, and we pioneered a new paradigm in structure-based drug discovery I'm going to talk about. And we're located in Marburg, Germany.
Fantastic. So maybe just tell us what it is you've got.
Yeah, sure. But first of all, I would like to ask everyone for 2 minutes of attention. I know we have full inboxes of emails. Just because we're solving decade-old problem in protein crystallography, I would like to share it with you. So there are two worlds that drive early stage small molecule drug discovery. It's the experimental part and the computational part we heard today. And the data that is driving both worlds, feeding them and amplifying, is structural data. Is where you see how molecules interact with the protein, what are the effects. And this is kind of day-to-day operations of pharma biotech companies to get this data, but the process to get this data is random in its nature. So it's trial and error. That's why in pharma companies, there are even fights for these resources because it's super valuable. But imagine what would happen if we would have at 10x fast access to this structural data. And this is what we do.
So tell me more. So where'd you get this data? How does it appear? And you know, how would I use it?
Great question. So the way to get this data is by means of protein crystallography, and protein crystals are super sensitive. They call it voodoo, magic. I don't know. It depending on the moon phase. So the consistency is reproducibility is very low. And nobody would have thought it would be possible. We have a rule-based process that stabilizes protein crystals by non-cross-linking in the native state. And this kind of expand the opportunity to test more reliable, and get higher hit rates. And because of the stability of the process, because of that, we utilize it as a primary screening, not as a validation step when we have something, but as a primary screening. It's the most sensitive method for screening. And the output is not binary, active, inactive, it's contextualized. You see what's happening and you can feed it into biology, medicinal chemistry, or combine it with computational capabilities, whatever you have.
So say I've got a kinase inhibitor and I want to test it. What would you do for me?
You have already an inhibitor, so you don't need that. If you have something, something like we are going we are reaching out to higher hanging fruits. So targets like RAS, MYC regulation, RNA translation, those are have been around for decades, but are undruggable. They say it's undruggable. For us, it's beginning to change, as we know. But with our screening, we have so far very consistent hit rates. They're higher. And with the data that you have, you can actually, what we heard from Shaha, there are different approaches like that. But it's the food for computational drug discovery. You can screen billions of molecules, and we have much shorter iteration cycles in drug design.
So what's in your inventive box? What have you invented?
So it was actually a observation. As I said, X-ray crystallography is perceived as super random, like there's no rules. It was an observation in the lab that something is occurring again and again. And it's the rules that we have, it's an experimental algorithm to stabilize protein crystals.
So it's a new way of doing crystallography.
Exactly. That's why crystals first. Everything is around.
How do you? I mean, your methodology is first in class, isn't it? Is it first in class? You're the only one with it.
That's a good idea to talk about. Yeah, it's first in class technology.
So first in class, but first in class is always a problem because you've got to, you have to get it adopted. So why would what benefits if I don't know, are you licensing it? Are you a service company? What model are you using?
It's actually a wonderful question. There are two issues. So the first one, we enter the market as a service provider just to validate in real cases. It works so far very, very nicely. And the first move, it's really for startup companies with this new paradigm. It was challenging. So we pitched this idea of screening primarily like as a primary screen on crystals. And the company said, whoa, we need affinity, right? But we stayed persistent. And last year, Boehringer published this paper where they go on RAS and say we should screen those targets. They call it X-ray first. It's same as crystals first. And they say that affinity biases decision-making in medicinal chemistry. So it's kind of a shift is happening in small molecule drug discovery, especially for those challenging targets.
So if I was head of R&D in a corporation and you wanted to sell it to me, and what would I get if I pay you money? Because I don't know a lot. I'm head of R&D, so all I want to know is what's it going to give me? What would I be getting by sending you a check?
New chemical matter for targets. What we heard today, there are targets where you don't have chemical matter, and we can unlock it. And the interesting part, that's a modeler perspective I bring in, that we would screen if our model is not predicting it will be a positive outcome.
So when and how will you get proof of concept?
What do you mean by proof of concept?
Like validation that your methodology works.
So we validated it on 20 plus, I think it's 30 now targets. We screen for pharma biotech companies on challenging targets. We are scientists, we're very cautious. We're still waiting for the exception, but so far it worked 100%.
But when you show it to pharma partners, what are they asking you to come back with?
When I show it to pharma, so they send me the targets. They say, okay, we need chemical matter for this target.
And but at the moment, you're looking still for a deal with someone. Is that correct?
So we are operating as a service provider. We raised funds. We raised 600K, similar to last presentation. We are active. We generate revenue, but the value. I'm just putting the company into therapeutics. So I want to shift the business model from service provider to a therapeutics company because there's so much to consider for our screening setup that in a service model we are limited. Because the biological input and a lot of data that is flowing does not come to us.
Right. And how much are you trying to raise, Sergey?
Currently, we have a pipeline of 4 targets, high-hanging fruits. We're raising 9.5+ million euro for Series A to kick off the execution of the strategy.
And have you got anyone cornerstoned yet?
What do we mean by cornerstone?
But have you got a lead investor or someone who's really up for it?
We started the process 2 weeks ago. We have a couple of conversations and we're waiting for the lead investor, but there are positive signs that I think in 2 weeks we have someone.
Fantastic. Now, time is up. That's a fabulous story. I'd
Great.
like to talk more and really understand what you've got, my friend. And let's pass it on the questions to Graham. Can you curate the questions?
Yeah, this time we'll go to Tom first. Age before beauty.
Okay.
I'm the
Right. Thank you very much. this is an absolutely fascinating area for, drug development, and, as you say, for the, determining, affinity of, molecules. But what differentiates your crystallography from, say, the Cairo crystallography?
So what differentiates it? The way we treat crystals is new. It's new. The industry standard consume it. The industry standard for the step that we're doing is trial and error. And we have like 24 hours procedure. We have stable crystals. We can screen directly. So that's the first one. And the second one, I think what the industry underestimates is that we've been working in this frame for 4 years. We have developed tools that solve this problem in a more efficient way.
Excellent. Next one, Graham. Graham, you're on mute.
Michelle, would you like to go next?
Yeah, sure. I'm still somewhat struggling here, I guess, because I understand you're doing protein crystallography to reveal the protein structure by itself, but you also talked about the molecular interaction with a potential hit or target molecule. If it's the latter, and you say like you provide new chemical matter, you actually need a library to pull out all the different molecules. Where do you source that from? And are you also looking at then the pharma company or another to provide a library, or are you hooking up, for example, with Shahar to actually provide here potential hits as an idea?
So we have access to proprietary fragment library. So we screen fragments directly on crystals. The exciting part is actually when you have the structure you have when you screen on crystals, and our technology delivers hit rates up to 30%. And the follow-ups also deliver 30% active, like you have 30% active compounds. The exciting part is that you start with multiple starting points. Let's say you have 10 fragment structures that you can start with, and then you can select for computational, like virtual space exploration, which one, and then qualify them and synthesize using enamines, capabilities, etc. So the first iteration cycle is very fast. It's 2 to 3 months. And we go consistently from high micro, low milli to low micro HTS-like hits within a short period of time, and add on, you have the structural data.
Great. Okay, if Michael, are you there?
Yep.
Great.
Hi. Okay. Yeah, I think I'm getting it. I'm just. Are you saying you're taking a sort of a fragment-based screening approach, but you've got your platform, proprietary platform, has a way without cross-linking to generate crystals more readily? Because I've working in drug discovery number of years, crystallization is an art form, it's a skill. So you're saying you've got a platform where you can knock it out of the park almost every time, and hence you can do fragment-based screening quite readily and get the structures no problem, and then build that information into a database that helps you pull out great hits.
That's a summary actually of my presentation. You start your process with structure, not from affinity. You start with a binding mode. It's a validated binding mode, and then you expand into chemical space using the fragment as a template with validated interactions. And in our cases, as I said, 30% hit rate is kind of the metric, whether you do SR by catalog, or you go into enamines. I don't know, they have now 17 billion molecules. Just you can synthesize or screen, not synthesize. We have 30% hit rate because we start with a validated hit, not with a signal. So just to add on this, the screening hypothesis in the industry is this. So you screen and then you narrow down, you do structure biology here. What we kind of turn it around. You start with structure biology, expand the chemical space virtually, and then you narrow down and synthesize the most promising and qualified compounds, not just doing series of compounds.
Fantastic. Okay.
We need to move on.
Well done, Sergey. That was very good. Thanks very much.
Really, really appreciate. Come back and tell us more. We love it. Let's move on to the next one. Now we're changing tack. We're going in a slightly different angle, and we're going to be talking to Elaine. Elaine, welcome to Coffee Buddies, and maybe you can just say who you are, what you do, and where you are.
Okay, sounds great. So my name's Elaine, and I run a pharmaceutical accelerator called Ascend Bioventures. I'm a protein chemist by training, but my PhD is in poop as a trained microbiologist as well. So, but really I've spent the last decade or so trying to find ways to make the drug development process faster and more efficient. I've been an inventor, I've been an investor, and I have served time in university technology transfer, so I look to find ways to help solve pain points in the early stages of drug development.
So tell us a little bit more about what it is that you do.
Yeah, because accelerator has a lot of different connotations.
Are you a company? Are you a confederate? What are you? And you know, what's the boundaries of your accelerator?
Well, I'm a human, and I'm located here in the Midwest. The pharmaceutical accelerator, we look for really interesting technologies and we form companies around them and make them ready for downstream partners. So we try to make that handoff between university to startup to pharmaceutical company as effective as possible, but we're virtual. And that was even before the pandemic, and not just because I'm an introvert and try not to interact with humans that much. We are virtual because we're located in the Midwest, and we find a lot of really interesting and overlooked opportunities in the Midwest, but being virtual, we get to leverage expertise from all over the world, and we don't make you come to Oklahoma. So that's, if you're scared of tornadoes, we don't make you come here. So. And being virtual allows us to spend money more on the science and not on office space and lab space. I've been a part of accelerators where I ran an animal facility, a medchem space and wet lab space, and it was a lot of overhead. And I prefer to spend money on the science part of it versus some coffee pots. So we also have an external advisory committee that has folks from large pharma, mid-sized pharma, and investment, and they help us make sure that we're picking the right deals. And I also have a parent company that they can serve as a co-investor in our rounds of funding.
So is it a bit like a translation vehicle? Is that what you've got, and you translate from if that's it, is it?
Yeah, a little bit. Yeah, I also liken it to fostering rescue dogs. I find interesting, promising technologies and try to shave the rough edges off of them and make them acceptable and ready to go for pharma.
And what do you see? I mean, you've got a portfolio, have you, of companies in it? Can you give us some flavors of what they are, sort of the sort of stage and what they do?
Yeah, so fortunately, I've been incredibly fortunate to work on 2 out of the 5 senses, and one of our portfolio companies is in hearing loss, and we have a clinical stage program that's already been licensed out and a preclinical gene therapy program we're in the middle of negotiations with a pharma partner on that program. That one is gene therapy. We are also getting ready to launch a new company called Recogene, which is a gene therapy delivery platform with a first indication in inherited form of blindness called Stargardt's disease. So it's really been amazing to work on something that could have such a huge impact on a patient's life and not make just iterative changes, but dramatic ones. And I also have several other technologies that are in the project management stage that I'm getting them ready to become a company.
And I mean, where do you use vehicles like Coffee Buddies to get out to talk to people, or how do you have a scouting team around your accelerator? How does it operate?
So yes, we scout for technologies, we get sent technologies. Having been in tech transfer, I work with a number of technology transfer offices, particularly here in the Midwest, where flyover as flyover can be. And so I leverage those relationships to find really interesting technologies and form companies, anything from an LLC to a C Corp, depending on what our investors like to do. And we find the very key experiments that pharma companies want to see or downstream investors want to see. So part of our diligence process is having conversations with Bayers and Mercks and Boehringer Ingelheims to say what what would make this interesting to you? And then we go do those experiments.
So you invest in your translation machine, take it to a value inflection point, and once you've got that, then you try and do it. So I mean, you know, I mean, one of your things must be how you get known, and I mean, how big is this, Elaine? Is it, I mean, are you talking about ticket sizes of 1 million, 500,000? What sizes of tickets are in your incubator or your accelerator?
Yeah, we're small. We've just been operational for about 1.5 years. Most of the investment rounds that we raise are around $1 million. Our first portfolio company was in the NASH space, and we were looking to solve the multifaceted problems with NASH. And we found that we did not. And we did that in less than 1 year and under budget. And we actually returned money to our investors because we found very key experiments that helped us get to that inflection point. $1 million is a challenging space to be in, though, in life science, because most life science investment firms like to come in at 3 million, 5 million minimum. And we are definitely at the seed stage. But again, I like to really find.
Are you before the dreaded, I hate this word, but are you before the valley of death, or are you in the valley of death?
I like to say that I'm in the shadow of the valley of death a lot of times. So I've taken on the later stage, but yes, shadow and valley. For sure.
You're prepping up companies so that they can get a better chance of not being in the valley of death.
Absolutely. I've been called sort of the Michael Clayton of startup companies. I'm an operations nerd, so I come in, I determine where the problem areas are, especially if it's a scientist-run company from the university side. I make sure that we have clean patent portfolios, the data that the pharmaceutical industry wants to see, in the right kind of models, and get them cleaned up for pharma and make that handoff easy.
What's your USP, Elaine, versus a TTO, a straightforward, a sort of a sophisticated TTO? Not that I'm saying TTOs are unsophisticated, but I mean, what I say, because some TTOs in universities have quite an elaborate group of which they can get funding into it, and they can translate things towards the valley of death. But what's the difference between you? Do you have stronger investors? Have you got more know-how? Have you got a better infrastructure?
Yeah, I think it's the better know-how and the experience, having transfer deals to large pharma companies and knowing what they require down to the nitty-gritties of diligence process. I work with tech transfer offices. I don't try to serve as the tech transfer office, but I definitely try to make that process easier for them and for pharma.
Fantastic. And are you looking for something? Are you looking for investment into your vehicle or investment into your companies? What's your look for?
Why am I here? Why are any of us here? Yeah. So obviously Recogene, as we're launching that company, is looking for funding. However, I'm wanting to pivot a little bit in my accelerator concept and find a strategic partner. Someone in the large pharma space that I can help identify new technologies and make that technology transfer process easier for them by partnering with Ascend. I want to look for pain points, and I want to look for gaps in white space and adapt to a partner's needs. So you see a lot of accelerators starting to partner with large pharma, and I'd like to be that partner. So it could be a possibility of joining with an investor group where we help seed those initial companies. But I think a pharma partner would be a lovely way to help connect the bridges and gaps in the valley of death, as we say.
And do you have a particular desire to make it global, or are you looking for a pharma partner that is, I don't know, focused outward around the globe? What's the sort of, is there, are you looking for a European partner because it gives you another angle? Is there any needs that you have?
For me, it's just more about the relationship. I want to find a partner where I can be of use to them, and vice versa. So to me, it's more about the relationship. So. Perfect.
Elaine, you've been a delight to talk to. Let's move on to the questions, and I'll ask Graham, the curator. I call him the curator.
Yes. Just because I'm curious and a bit curious.
Get some questions from our friend.
Michelle, we'll start with you this time. Thank you.
Okay, no, thanks, Graham, and thanks, Elaine. Very interesting. I'm not sure we'll become that pharma partner. I can just not comment on that. You'll have to discuss it with my US colleagues. But what I'm quite intrigued in is I hear NASH, I hear hearing loss, I hear gene therapy. Do you actually have a sort of a sweet spot that you're looking for, like specific expertise? I know myself how difficult it is to switch between therapeutic areas all the time, but I'm just curious on your experience there.
Yeah, I definitely have areas I try to stay away from, just from past experiences. So that's more of my area. But I look for white space, definitely. I mean, so hearing loss was one of those areas, and then gene therapy delivery was one of those areas, and women's health is another area that I'm interested in, because there's just NASH was challenging because there were a ton of people in NASH, and so in looking for the white space, and that was really difficult. We were looking at it more from a fibrosis perspective because that was the white space in NASH, but yeah, really trying to find the gaps and then pulling the teams. Like you say, it's hard to be the expert in all the things, right? So you want to pull the teams around that have that expertise, and that's the benefit of being virtual too.
And where do the experiments take place once you sort of selected a scientific lab? Do they still perform at the university where the teams most likely are already hosted? And how do you deal with that? Do you then pay for overheads there? And just out of curiosity in the business model.
Yeah, so it depends on the asset. So for the hearing loss company, for example, there's not a lot of CROs that do hearing loss. So we were very much tied to the research institute. It wasn't a university, it was a research institute. So we did have to deal a little bit with overhead. But I try to find partners that are in the CRO space that can help, one, provide third-party validation, but also get it out of the university lab, pay a little less overhead, but also, yeah, the third-party validation is really important. But it depends on the asset and the specialty. So.
Thank you.
Great, fantastic. We'll go for Michael next, please.
Hi, Elaine. Thanks so much for taking us through it. Really exciting and what you're building there. I just wondered, Michelle asked you about different indications, and do you go firstly for with your network of, say, institutions, tech transfer offices? Do you more or less sift there and then find opportunities by what's on the table, or do you actively go out for specific indications? And do you tend to focus more in, say, Oklahoma and the region around that, or do you go further afield?
So it's a bit of a combination of all those things. I get a lot of things passively. So if people send me deals, they know what I'm interested in and they'll send me things. And I certainly have relationships with different tech transfer offices that they'll send me things. I have partners here in Oklahoma that I see everything in their portfolio and I help advise them on that. The hearing loss company, for example, was here in Oklahoma. So sometimes it's lucky to have it in your own backyard, but the Vision company, gene therapy delivery company, is actually from Case Western University, which is also in the Midwest. But my NASH company came from San Francisco. So it's a combination of all those things. Really, it's finding the right opportunity, and some things I have to actively look for. Women's health, it's been very challenging to find things in women's health. And so I'm actively looking there, but I do receive a lot of deals just passively.
And has it been easy to find, so you find these opportunities, maybe the founders, the PIs, academics who have developed it, is it then easy to find, the drivers, maybe the C-suite or those experts who have, built companies before that you can bring on board, or do you find that a bottleneck?
It can be a bottleneck depending on the indication, but I actually partner with another company called Smart Health, and they have a cadre, a stable of biopharmaceutical executives being located in Chicago that are interested in sharing their time. But it's actually a fairly easy thing to find people that are interested in opportunities, especially when we can be so flexible as to, you don't have to be the full-time CEO. You don't have to come to Oklahoma. So we try to make it as easy as possible to make the experts want to come work for us.
Yeah, no, in Midwest and Oklahoma, there's so many great opportunities and everything else. So it's really great what you're building here. And I'm sure there'd be some really good, when you're thinking East Coast, West Coast, and that focus inherently is there, that you'll find some gems where you are.
I hear Oklahoma's very nice as well. But Tom, you're next.
Yeah.
Yeah, Hi, Elaine. Conceptually, what you're doing, is something I started in 2007 when the word virtual, was a bad word. but now, you're in an ideal sweet spot,
Yep.
and I've worked, with, tech transfer offices, particularly here in the UK, but, my way was going into big pharma, and trying to pick out compounds, some of which I developed off the shelf.
Mm-hmm.
But I think, you're in a great space, and, I wish you luck, but is it just you?
It's just me, except that I have a parent company. So I have 4 consultants that I use to help me with the daily operations. I have a parent company that helps me with all of the other operations, and then I pull in expertise per the asset. Yeah. So that was a lesson learned from a previous accelerator, is that we don't have to be all the things for all the companies. We can pull people in as necessary. But yeah, the key operations, no, we have people for that, thank God, because I hate being an accountant. It's terrible. So.
Well, I think we need to move on now. First, massive thanks to Elaine. That was super dupe, and we look forward, Elaine, to seeing you next time. And maybe you could bring your companies. So, we'd really appreciate seeing your companies and the entrepreneurs connected. Now we're going to move on to the next one.
Trevor, yes, Trevor Castor.
Again. We're going to Trevor, and welcome to Trevor. Trevor, a great delight to see you, my friend. And maybe you could tell us who you are, what you do, and where you are.
Thanks, Tony and Graham, and great portfolio presentations. My name is Trevor Castor. I'm President and CEO of Atheos International. We are based in Woburn, Massachusetts, United States. We are developing the next generation of pathogen reduction technology for manufacturing of virus-free plasma and plasma products, biologics, recombinant biologics such as therapeutics and monoclonal antibodies, and medical devices such as PPEs, 495 masks, and single-use devices. Well, there's an obvious reason why we need a technology like this in the worldwide pandemic caused by viruses. We're trying to sort of put a break on this virus, as well as put a break on future viruses. There is a problem with virus, and in terms of transmission, in terms of deforestation, in terms of rapid plane transportation. And we have new viruses that come up, which we don't know what classes they are. Typically, we in this technology area categorize viruses in 2 classes: enveloped or fatty encased viruses such as HIV, coronavirus, influenza virus, and non-enveloped viruses, which are protein encased viruses such as parvovirus B19, hepatitis A, poliovirus. So most of the technologies out in the marketplace today only inactivate envelope viruses. They don't inactivate a second class of viruses. Our technology works in a different manner. It's unique. It's a physical technology. If any of you are scuba divers or free divers, you'll learn that always rise slower than the bubbles that you generate. So that's what we do. We fill the viral particle up with a fluid, and then we rapidly expand it and pop it at your weakest point. Therefore, the technology is generally applicable to both envelope and non-envelope viruses. So, and it's physical, so there's very little impact on the biological activity of the substrate that we're working with. So that's a significant advantage versus the chemical techniques which are out there, the heat techniques, the solvent detergent techniques which are out there to
Trevor,
minimize transmission risk.
So Trevor, I mean, how did you get into this area? What was the journey to get what you've got?
That's interesting. Our journey started with trying to develop a Trojan horse process for recovering recombinant proteins and enzymes from yeast. Yeast is a very hardy eukaryote macroorganisms. And because it's a eukaryote, it can produce proteins which are fully glycosylated versus E. coli. Most of the industry went to E. coli because it's easier to get the proteins out. So yeast, you have to basically break it with a French press. You have to go to 25 to 30,000 PSIG, and you have to compress it and break it. And we said, well, there's a better way of doing it. Can we get into the yeast particle and expand and break it from the inside out? And we were able to do that very effectively. We were not able to translate that into biologics companies having focus and use as a mechanism of creating recombinant therapeutics. So we changed our target to viruses. And the viruses are much smaller than yeast. So will it work? We found that it will work to the nanometer level. And can it work without damaging biologics? And we found that it will work without damaging biologics. And that was our history or transition from straight biologics to blood plasma and biologic products.
So what is it that you do with, is it a device? Is it a chemical? What is it and what does it do? What's the actual invention?
It's a combination of device, technology, and supercritical fluids. So the working fluid we call them SFS or superfluids. They are fluids which, when we compress them, exhibit enhanced thermodynamic properties of penetration, solvation, selection, and expansion. So we expand the fluid. So the fluids that we use a combination of fluids which will expand and break the virus but not damage the proteins.
So is it going to be in a process system? You
Right.
may have a device that's attached, a bit like in the oil industry, they have attached a piece of chunk of metal or something as something goes down a pipe. So you've got a device that does something to the biological fluid that is going through it. Is that correct?
So let me explain it a little bit easier for you. We have an isobaric chamber, which can be this high and this wide, which we have the SFS in the isobaric chamber. And then we inject the biologic or plasma that has viruses into the isobaric chamber. So we inject them in small droplets in the micron size or smaller. So the virus will reside on the surface of the droplet. It will contact the SFS and be saturated by the SFS. In 20 seconds, it will drop to the bottom of the chamber and coalesce into a pool, which is then removed from that chamber. In that 20 seconds, we're able to saturate up to 1 million particles of virus per milliliter. Once it comes out of what is a back pressure regulator, it expands and the virus is interrupted. The nice part about that, Tony, is that we can go to different chambers. We're going to stage in it. So we can go from one isobaric chamber to second isobaric chamber. So in 5 isobaric chamber, we theoretically can get 30 logs of inactivation of most envelope and non-envelope viruses. However, we have designed the system to have at least minimum of 1 log per stage, so that we'll have a minimum of 5 logs of inactivation, which is a requirement by the FDA and the EMA.
So you're looking for money and you're looking for customers, I presume.
Yes.
And if you're looking for money, how much are you looking for to do what with? And if you're looking for customers, what's the persona of your ideal customer?
Okay, we're looking to answer your question specifically. We are raising $5 million to develop data site. We have very extensive labs here in Woburn, BSL-2 and BSL-3 labs. We have several alpha site prototypes. We're looking to build beta site prototypes and continue our discussions with the FDA to start the process of getting a PMA. And part of that process is that we have to do clinical trials on as a CFI treated product to get approval by the FDA. So that sort of dovetails with partnerships. So Pfizer has evaluated our technology, and in the Andover facility for biologics, they like the technology. They're waiting for us to have FDA approval of the technology. But we're working with a small company in Atlanta, Georgia called Cambium Technology, which is developing a dry eye formula made of human lysates for dry eye. So we are going to piggyback with that company to take our process of demonstrating pathogen inactivation of their product to get FDA approval. It becomes our flagship, therefore, to say, okay, we have FDA approval of a clinical trial product. Let's go on to Pfizer, other collaborators who may need our technology.
Fantastic. Now, Trevor, I'm mindful of time. I've just had the great tea come up, and we could talk a lot. I mean, I think I'm getting the picture here. Fantastic. Sounds awesome. Can I just bring in Graham to ask her a couple of questions from our pharma friends?
Yes, we'll go to Michael first.
Hi, Trevor. Thanks so much for taking us through that. Really interesting. I just wanted to know, how did it all start?
Well, actually, Aphios is a combination of two Greek words which mean virus-free. And that was provided for one of my good friends and buddies in Ilan Papadopoulos, who resides in Greece now. So I'm not going to take credit for it. So we started looking at how do we provide, I'm a chemical engineer and a mechanical engineer. How do we provide my skill set to enable the biotechnology industry to expand and to grow? So we looked at drug discovery, drug manufacturing, nanotechnology, drug delivery, and pathogenic drug safety. And we looked at them as unit operations, as chemical engineers tend to do, and then look at what are the deficits, what are the problems, how can we address the problems, how can we increase manufacturing, how can we improve in terms of both drug discovery or manufacturing or pathogenic drug safety. And that's how I started.
That's great. Well, thank you.
Right.
Michelle.
Michelle, yeah.
Yeah, thanks, Trevor. That was helpful indeed. I'm just very curious, where do you really see your sweet spot? Because I am, for example, thinking of biological manufacturing processes where viruses are used to transfect cells, for example, that eventually you need to remove. But I don't really understand if that's a sweet spot, or do you see somewhere else? Where exactly in the manufacturing process, if I understand it correctly, do you really place this?
Do you remember a few years ago, a Genzyme facility got shut down because of virus contamination? So that's a sweet spot where all biologics are required to have a virus clearance method. Mm-hmm. But you want to have a virus clearance method that does not damage your valuable proteins or reduce your valuable proteins. So you want something that can be very physical, normal to your viruses, but not damage your proteins. If you do nanofiltration, for example, you have a high loss. For example, in manufacturing immunoglobulins, very costly. Loss of the product is a significant problem in that. So that's a sweet spot, for example.
Right. Okay, so really at the end of the drug substance manufacturing in the sterilization process. Yeah, all right, excellent. That helps a lot. Thank you so much.
Last question. Thank you, Michelle. Tom.
Yes, Tom. Oh, you're on mute, of course.
But cost. Cost is always important. So
the dog was barking. Anyway, great discussion and talk. Back in the day, we used to use formaldehyde, to bomb sterile facilities where, you were making smallpox vaccine, etc. But, this is a sort of novel, relatively novel technique that you've developed. But the scalability with regard to what we're seeing in the world today, and to relate to Michelle's question, we have manufacturing problems in the world with regard to vaccines. So how scalable is this technology?
It's based on engineering principles. Liquid operations is quite scalable. When you look at engineering principles and you look at liquid solid or solid solid, you run into scalability issues. So you have to think of a chromatographic system where you can scale by improving the cross-sectional area for the flow rates. We have scaled it to 10,000 liters per day, and we've scaled it down to operating at 200 mils per unit. So we've gone through the entire scaling process. I think we are better off on the high scale right now than on the unit scale, which we are still working out some kinks in it, in the unit scale, where we have to work with 1 unit of plasma. But for the larger scale, we have built a pilot plant here at Aphios, where we can work at about 1,000 liters per day in an 8-hour shift.
Tremendous.
I'm mindful of time. Trevor.
Yes, that was excellent.
Thank you, Trevor.
Trevor, thank you very much. Round of applause.
Great program, Tony. Great program.
To come back
Thank
and tell us more, because it's fascinating. I like that stuff. So thank you very much, Trevor. Let's move on to, we're changing tack yet again.
Yes, something else novel.
Hakim, where's Hakim on my screen? Let me have a look.
He is there.
Are you
Hey,
waiting?
Tony.
There you are. Fine, I found you. Fantastic. Hakim, welcome, my friend. And maybe you could introduce yourself, who you are, where you are, and what you do.
Yeah, thank you, Tony. Thank you, Graham. Great to be here and great to listen to these talks. So I'm Hakim. I'm one of the co-founders and CEO of Closed Loop Medicine. We are a UK-based healthcare company, and what we are focused on is developing healthcare solutions that bring the very best of what we're learning from the application of digital health and digital therapeutics together with traditional medications. And so we at Closed Loop Medicine are developing new combination products of drug plus software as a medical device, with the aim that a combination product can be prescribed from a single prescription. So a physician can prescribe and a patient can receive an integrated care package of drug and non-drug therapy in a simplistic way off formulary. I came from a background of working in the health system and saw the fragmented nature of the individual therapeutic modalities, whether they be drug, digital, device. And actually, if we're ever to help patients get to the outcome they need, we need to start thinking about how we optimize these different modalities alongside one another, really building up a greater picture of the patient journey from data from devices and diagnostics, from medication and regimen, and also now the value add that we can gain through the real world insights generated from digital health and the provision of care through digital therapeutics. So over the past few years, we've been developing a team that can develop drugs and software as a medical device under one roof.
So this is the word closed loop. That's where that word comes from. I mean, just Hakim, we've been, for a long time, I'm a rheumatology person, so we've gone back. We would always give a non-steroidal with a steroid, and then we might give a bit of gold or something. So combination therapy is not unusual. And so what's the twist you've got with the combination and the software? Tell us more.
No, absolutely. So combination therapies is something that we've been doing for decades. Since the beginning of medicine, the advice that you get from your clinician and then the prescription, or maybe it's a different mix of therapeutics. What we're doing here is bringing them together under a single prescription to make it easier for the physician to prescribe and the patient to receive both the drug therapy and then the digital therapy, whether that be behavioral wellness, lifestyle, diet and exercise, but using the real world data to start to optimize the amount and time of that medication, whether it's getting a patient to an optimum, whether it's weaning off a particular therapy. And whilst we've had a number of drug device combinations, drug software as a medical device combination seems to be a novelty, and through the engagements that we're having with the regulators, where we're seeing a really great feedback from them in wanting to work with us in bringing something that they believe will have greater benefit for patients by having this integration of addressing both the drug and non-drug therapy. And as I say, where we've got to is we've built up a team in-house in the UK where we're developing our own drugs. We're not doing any drug discovery, so we're working only with known drugs, changing and modifying formulation, building the appropriate software as a medical device to support that drug, and taking that through clinical development. We've got 2 programs in development, one in insomnia and one in hypertension, neither of which on the market yet, as I said, still going through clinical development, but trying to bring something that provides a more holistic and integrated care solution for patients and physicians.
So if you take your cardiovascular, can you give us some idea of what, so you may have, I don't know, a few different drugs for your heart, blood pressure and everything. And then what am I going to be, is there when I get a prescription, will it say on the prescription, a bit of software, which or a mobile phone, what will be on the prescription? And then as a patient, what would I get? Would I get a mobile phone and a set of tablets? What would I get?
So the way in which we're approaching this at the moment is to really think about the gateway drugs, where patients are likely to be prescribed a first in-line therapy. So at the moment, we're working on a single drug, single software as a medical device combination, but very much aim with the fullness of time to build out the opportunity to have multiple drugs, because particularly in areas like hypertension, where you would expect to have multiple therapies. What we're doing is taking that drug digital combination through clinical trials and regulation. We built a single quality management system, allowing us to co-develop drug and device through a market authorization application, so that we'd effectively be on formulary. So yes, the physician only would be writing a prescription. On that prescription, you'd be getting a link to access the software as a medical device. And there are different ways of doing that, very much dependent on the digital maturity of the health system.
If you take something like the CNS area, and Tom's our resident CNS expert, so it'd be interesting. I mean, CNS is clearly something that depression is clearly something where an app is being prescribed, along with yoga, by the way. And I do yoga, and you're all welcome to join my yoga events. But I mean, so you have your app, and then you have, you've got your tablet. So just tell us a little bit more about what you've got a link. So what will happen is I'll go home, take my tablet, and then I'll link myself into the computer, and it will be bespoke for me, because so it will have a link with a number on, which is attached to myself, which gives me an independent data system that connects my data into that. And my question about data privacy is an obvious one as well. Who's data?
So the first thing we've done is sit down and work with patients and patient groups to understand how they want to use this type of technology, how they want to access it, and talk through all of the appropriate data concerns. So in a situation that you describe, you might pick up your prescription. If it's in the UK, where many people's mobile phone numbers are linked to their EHR, you could receive a text on your phone to download the app directly from your prescription. That's something that is already happening in the UK. In other health systems, there'll be other ways of doing it. Because we are owning both the drug and the software as a medical device, and we own the packaging of that drug, we've got opportunities, whether it be through QR code or otherwise, to provide that link between the drug and the app. You're then opening up the app and building out your own healthcare record supporting that particular drug use, whether it be the timing, the amount, accessing the lessons, the behavioral content, and building out that area under the curve that so rarely gets understood between clinician visits, to better inform changes to the therapy and to use the clinician insight to make those changes going forward. We very much see this as a platform to product opportunity, and we've already identified a range of different areas, and CNS is an obvious one where this would have application. We're working as an early stage company in the first two indications in insomnia and hypertension, but see the value of applying this to other chronic disease settings. And in terms of opportunities, we have the opportunity for taking our own drug digital combinations and working with some of the very forward-thinking specialty generics. But equally, we're now starting to get approached by specialty pharma, specialty generics and pharma to ask us how we can close the loop on their drug. And so we're now starting to think through interesting scenarios of how we take this approach from an IP regulatory development perspective and applying it to other people's drugs, not just our own.
Fantastic. So you're looking for money and you're looking for partners, I presume. Hakim,
Tony, I'm a CEO. I'm always looking for money.
Well, no, absolutely. Unless you're in some places when you're getting abundant. Anyway, back to that. So how much you're looking for, and what's the persona of the person from the pharma company you're wanting?
We've had a fantastic support from our seed investors. We've got 3 institutional funds and the Cambridge Angels supporting us at the moment, 2 tech funds and BGF. And we're now going towards our Series A, where we're looking to raise our next substantive amount of funding to take us through to the next value inflection point for the company, which is to take one of these drug digital combinations through market authorization and put it into the hands of patients once been prescribed, other than the testing we've already done. the persona you ask is really fascinating. We sit right at the middle of a variety of different investment theses: the traditional life science asset-centric model, the tech model that is moving closer towards the life sciences and regulated and healthcare. And for some traditional life science investors, we're too digital and doing generics. For tech investors, where the fear of being regulated is not something they want to step towards. And then the emerging world of that convergent landscape. So for us, the persona is really about understanding the convergent landscape of life science and tech, which I know you've called in the past tech bio rather than biotech.
Well, I think in a way you're even tech clin, which is even more interesting. So I'm going to open it. Hakim, you're always a pleasure to talk to, and thank you so much. I'm going to ask. I'm going to first of all bring out our CNS man, because I've mentioned his name, and so Tom, maybe questions for Hakim.
Yeah, Hakim, this is obviously the future, and how it integrates, with various therapeutic areas is going to be, amazing in some cases, but disastrous in others. But one of the things I have concerns about is the sort of lag time, between the digital feedback, with regard to a patient. So take for example insomnia, where the current treatment is still sort of benzodiazepine-like compounds, and you're treating these people over a period of time, and often it is the case where the patient has to come off the benzodiazepines, and then there's all kinds of sort of rebound effects. So the type of information you would get from sleep parameters of a night with your Apple Watch or wherever, great, you can see, but where you're actually prescribing a drug which can have adverse effects, that lag time is so important in getting that feedback to the primary care physician and changing the treatment. And I think that's my concern, because even today, and I know COVID is overhanging everything, it's still very difficult to get back to your primary care physician to change your prescription.
No, absolutely, Tony, and that's why we've been very careful in the drugs that we've chosen to begin with, in terms of that safety and adverse event profile, for that very reason. And the guidance that we're following is very much the one that's been dictated and coming out of the FDA, in that in the first instance, these types of technologies should only really be used to inform clinical decision making. So the clinician is still in the loop. As you mature and start to build a greater understanding of these technologies, you start to make recommendations on those changes, whether it be coming on or off, or changing the dose, and ultimately getting to a point where you can automate, and hence the name closed loop, where we get to that model. The guidance that we've had from the regulators so far are that there is a level of comfort in working within the existing known dose ranges, but certainly going above or beyond those is not something they're willing to take any risk on, where we're putting patient safety first and foremost. So we've started with drugs where there is a much greater safety profile to be able to build this out. And you're right, this is one of my good old colleagues from my past consulting days used to say that platform technologies are dangerous. They're a bit like a baby with a hammer. Everything looks like a nail. And we have to be really quite careful which indications that we choose here, because it's not going to be appropriate for all.
Fantastic. Next question. I'm mindful of time, Graham, because we've got two more.
Yeah, we're good. Yeah, Michelle.
Michelle.
Okay, yeah, I had two questions, but I'll limit to one. So if I think about digital therapeutics, a very high topic, or hot topic, I should say. So great work. But what really differentiates you from companies, for example, like Sidekick Health, who does pretty much the same thing? Maybe they have a different partnership model. And what is actually the big hurdle for, let's say, copycats? Is there IP, or is there data exclusivity you generate? How should I see it as protective?
No, great question. So Sidekick, Pear, Click, Achille, any of those digital therapeutics companies, none of them are developing their own drugs. So we are developing our own formulation of drug and the app, where we put both through clinical development and both on the same prescription. Now, a number of these digital therapeutics companies are forming, as you're very well aware, strategic partnerships with pharma. This, and sitting alongside as a companion, this is a fully integrated. And as part of that integration, there's both regulatory and IP protection, both on the platform and how we bring the two together. Be happy to pick it up offline if there's an interest.
It was actually my original question, like, why do you want your own reformulated own drugs? You talked about packaging with QR codes, which you can do as a generic as well. And I don't really see the added value for an investor to fund that part as well. But you say that otherwise you cannot link it up into one prescription if you don't own it both, is it?
You can link it, but there's a deeper level of IP that we can get that has greater value long-term for patients. By being able to reformulate and think about novelties on getting that dose right for the individual.
Great, and a quick one from you, Michael.
Yeah, very quick. Thanks ever so much, Hakim. Just a quick question on data. You know, you have the app there. You'll be pulling together a lot of real data that would be lost from clinicians and carers alike. So what are your plans on data? Because I'm sure that would have been thought out from the get-go in terms of how you monetize that aspect, but give back in terms of information that drug companies and the like could benefit off.
I think the key thing here is that level of integrated, longitudinal and personalized. What I saw firsthand working in the health system is that, well, Activa had the pharma data, the digital health companies had the PROMs, and the medical device companies had their data, but there was a very, very difficult way of bringing that together, either at the level of a patient or a population. And so ultimately, long term, by doing what we're doing, we very much hope to be able to curate longitudinal integrated care records that can provide better insights in terms of population health management for health systems, better insights in terms of utilization and optimizations for pharma, and better insights in terms of being able to deliver better care from a physician perspective, but ultimately to empower patients to have that single record. So for us, that long-term data strategy really sits at the heart of why we set up the company. But thank you for the question.
And would you like to?
Fantastic. I think we're going to have to move on to that because I think we love it. We love it. It's fantastic. But first of all, Hakim, fantastic, man. Come.
Thank you. Thank you, everyone.
We really appreciate what you're doing. I think it's great for patients, and we're looking forward to watching you go from strength to strength. So we're going to move on to the next one. We're changing tack again, aren't we?
Katia, yes.
Katia, welcome, Katia, to our Coffee Buddies event, and maybe you can tell us who you are and what you do.
Hi, Tony, and thank you, Graham, for inviting me. So, first of all, I'd like to say thank you, Tony, for revealing that you're also a yogi. I'm also a yoga practitioner. I'm actually a certified yoga teacher.
And me, fantastic.
So so a little bit about myself. So I'm this is my second company. So I had a company called Apretica, run from 2006 to 2010. Actually, I'm happy to see that one of my former customers is on that call. One of the presenters, Trevor Castor, he did some work with AFIOS, did some work with Apretica early on. So it was a discovery CRO. So my expertise is in ADME, predictive toxicology, de-risking drug discovery, mostly small molecule programs. So I'm also a proud mother of a recently graduated registered nurse.
Ah, fantastic. Tell us more about what you're doing. Tell us more, embellish it a bit.
I am an academic, so I recently, last 6 years I've spent, thank you, Lynn, in academia at Johns Hopkins University, and I've come back to the drug discovery space just in the last year. So we formed Activa Therapeutics at the end of last year, so we're very new, but we have filed provisional IP and have an option from a university to go forward with this. And thus far we have, in terms of the funding, we have founder funding. So what do we do? So Activa Therapeutics is focused on activating the kinases, on activating molecules that traditionally have been inhibited for their biological function. And the reason we do that is because one of the founders has found in his research that actually activation of one of the kinase pathways leads to a repair of endothelial vascular barrier leak. And that's the core of what the molecules that we're developing are for repair of vascular leak. And our first indication is we're addressing this in the lung, which is ARDS, acute respiratory distress syndrome, or acute lung injury that potentially could lead to ARDS.
So tell me more about what you mean by activation. I mean, what is your activator, and how are you activating?
Yes, so happy to tell about that. So basically, you have kinases that typically there's a few many cascades that phosphorylate each other, right? One kinase means it phosphorylates something else. So, and traditionally the biological, for a variety of reasons, for knockout models and things, the first thing that people would do, they would try to knock out a particular member of the cascade, or knock out a gene and see what the phenotype is, right? So we found that actually in this case, the research started with inhibiting that protein, and it turned out that actually the results were the opposite. That actually the results were opposite of what we expected. So what we did when we developed an activator that binds to the kinase and actually keeps the protein phosphorylated instead of being unphosphorylated. If you inhibit it, you prevent the phosphorylation. So in our case, keeping the protein phosphorylated actually is the biological function that we want to preserve in this case.
And what does the biological function do then? You've got a phosphorylated protein. What does it hit a receptor? What's it? So it's a kinase.
It's an intracellular cascade downstream from P38, which
okay.
is a typical target.
So you end up with a. I mean, these activators and inhibitors always intrigues me. And what do people say to you when you go out and present this? Is this a novel idea? I've seen it with other modalities, but what do people say about this whole activator story?
Yeah, so it's very situational. I think we all appreciate here that biology is very complex. So in some situations, some targets should be inhibited. In other situations, that should be activated. And once you start going into that, the story becomes really very complicated. So the interesting thing here is that pathway has been tried in clinical trials for inhibition multiple times, and all clinical trials actually failed.
And what would be your proof of concept? So maybe you could talk through what a proof of concept experiment looks like.
Yeah. So there are some animal data that we have with the peptide molecule that we have licensed out of the university where original work was done by one of the co-founders. So in the lung model, the activator molecule and phosphorylation of that target actually prevented the vascular leak. We also have the in vitro evidence of that phosphorylation being activated by this peptide and other molecules that we have developed since. So in the last year, we have developed a way to discover small molecules that instead of the peptide that bind to the same pocket as the peptide on the target. So and that's where our technology for discovering the molecules comes in. So we have discovered this technology to work on that target. But as we have matured the technology and started discussing it with various potential partners, we found that there are other things that we could do with that technology. So it's based on published research of one of our co-founders, and it is AI-assisted machine learning-based molecular docking program that we have discovered. But we are not mining billions of molecules. We're actually our starting point is approved drugs with ADMET filters that actually are predicted to not cause anything horrendous. And it's a prediction, right? But it's prediction based on our expertise of our team and based on approved drugs, right? So that's the starting point. And then we optimize these molecules to fit the same pocket, and also have characteristics that we need from our target product profile that we develop for each indication. So in this case, we're working on the ARDS. We have other conditions that we are considering in the pipeline. The vascular leak is underlying over 60 medical conditions. So we also are talking to some partners about developing a combination therapy for vascular leak. Is a side effect of some medications, especially monoclonal antibody-based chemotherapies, oncology drugs. So we're talking about some of those things.
Interesting. So, so what what's your strategy going to be moving forward? You're going to try and outsource out license early? Are you going? And how much money are you looking for? How much money have you had so far? My question, I suppose, I'm still trying to get my head around this activator concept. So, how much money have you had? How long have the patents for this sort of thing, or how long has this invention been around? Is it a new invention, or is it something that's been known about a long time?
So the biology is in public domain, right? So it's not patentable. We're just exploiting this for this purpose. The patents are around composition of matter of the structures and methods of use and formulations. So that's really the IP. Another piece that's sort of secret sauce is that the way we discover the molecules, that based on our knowledge and some publications on cheminformatics.
So has it been, was the original discovery some time ago?
Yeah, it's about a decade ago.
So why have people been sitting on it for quite a long time and it hasn't sort of come out?
Well, that's a good question. It's a loss of funding, loss of NIH funding, and the team basically no longer worked on that. So that was one of the major reasons. And
are you are you a fully fledged company with people in it, or are you virtual at the moment?
We have a lab where we conduct experiments, and we are selecting the clinical candidates. So we have 5 people, the 4 co-founders, and one consultant who are working on this for a year.
We've got to think about moving on to questions now. Thanks very much, Katia.
Ah, good. Graham, that was a rapid entry.
I know, you can tell you're intrigued, you know.
I am, because I was trying to get Katia, fantastic. I got stuck in, I'm trying to work it out actually. So let me just answer the questions.
Michael, we'll go with Michael first.
And I can take questions on yoga as well.
Oh, good. What's a vinyasa? Okay, on to Michael.
Hi, Katia. Thanks so much. Really interesting. Could I just ask quickly? Is it a peptide in terms of the focus that you're developing, the machine learning aspects of that, or are you using other modalities?
Peptide was a starting point. So this was the original discovery at the university, but we are developing. We are focused on small molecules.
Molecules. And are you going basically for undruggable targets in particular, or is it all on the vascular side?
No, it is a druggable target. It's been used in the clinic unsuccessfully.
Yeah,
we think it was inhibited.
I mean,
it is a druggable target.
I mean, is this are you going more a single asset side, or is it a platform with the machine learning AI side of things to go after a whole range of other targets?
Yeah, we have other targets in mind in that pathway, and also other synergistic targets and other pathways that also can work on vascular disorders.
And is it that you're looking, because these are what you're going for is a drug that's already been developed that well, but you found another use? Is your platform the strategy to look at other molecules that have been used, and in effect the readout wasn't that good, to find it almost a repurposed play? and from there build on to develop other assets?
Yeah, that's a good question. We do have a starting point also. So we have sort of a peptide repurposed drugs, NCEs sequence. So we do have some drugs that we are considering that are repurposed, but at the same time, we're developing NCEs as well. Repurposed drugs have their advantages and disadvantages. One is we can get to clinical proof of concept relatively fast, but it's limited IP protection. So we are working on NCEs as we are doing the repurposed at the same time.
Right, so we'll move to Michelle.
Yes, thank you for that, Katja. So there are some activators of kinases known, of course, but the majority of information we have is from the inhibitor space. And nearly all of them have really severe side effects and a lot of black box warnings as well. So in basic principle thinking for me is tampering with the kinase system is a very, very risky business. So how are you approaching de-risking this strategy?
Yeah, so we have a bunch of filters that are already built into design of our molecules. And we also have a very robust de-risking ADME and investigative toxicology suite of assays, which is my expertise for the last 15 years on how to de-risk them. We have, I'm very aware of the profile of the kinase inhibitors, and we have them as controls in all of the assays that we're putting together. So it's experimental. It's both in silico prediction and experimental approach.
Fantastic.
Tom?
Tom.
Quick, take yourself off mute, Tom.
Again, yeah. Yeah, this is a, fascinating area, and, an area which I have some interest in, from how, you can enhance the efficacy of a, receptor. And, the pleiotropic effects that, Michelle discussed with the kinase is interesting. But one of the hot areas now is the promoter methylation induced epigenetic silencing of enzymes. And I was wondering if, this is a sort of avenue that you're following.
We're not pursuing it as we speak right now, but it's not technology that we have pursued. But it certainly is an interesting area. Again, we are in our experiments. We show that it's not silencing; it's actually putting loudspeaker to those kinases.
Yes, yeah,
is working better.
Fantastic. Right. Well, I'm going to move on to our last one in this amazing.
Thank you, Katja. Brilliant.
Lovely to see you, Katja. We'll see each other on a mat, I hope. Let's move on to Mike. Mike.
Hey.
How are you, Mike? Oh, there's Mike in the Hawaii.
I'm here.
He's a man who's living the dream.
Yes, if only.
Mike, can you tell everyone who you are, what you do, and where you are?
Yeah, sure. Well, believe it or not, I'm actually in sunny Manchester. Now, the sunshine in Manchester this morning was just like you see behind me, but true to Manchester, it's actually throwing it down with the rain at the moment. But there you go. But I like the sunshine side of things. So this is what I am. So I'm Mike Davis, Dr. Mike Davis. I'm the CEO and founder of a startup company called Carousel Bio. And we're developing novel peptides with nanoparticle delivery that we're using to switch off inflammation. Now, we do this by targeting something that Katja mentioned called MAP kinase, P38 alpha. And that's the orchestrator of inflammation. And we switch this off at allosterically binding away from the known sites of binding. And we've demonstrated that we can not only switch off inflammation, we can prevent the activation of inflammation. So we're early. There's lots of areas that we could go into. We're sort of targeting skin diseases, first of all, because you'll be able to see it more effectively. And we've had an Innovate UK grant, which is the same as an NIH, but it's in the UK. It's an Innovate UK grant, and we've had a little bit of seed investment. So we are at the stage now of starting our studies looking at human tissue biopsies and seeing that our compounds work in human tissue biopsies.
So Mike, I mean, I love your description, MAP kinase. I mean, there's this phrase now called guilty targets, and so MAP, you said it so powerfully that MAP kinase is it, and you know, I don't know about that, my friend, but let's hear more about that. MAP kinase is in the center. What do your peptides do with the MAP kinase? So
basically, the peptide, we've got 5 different peptides. The lead peptide and the others will work in the same way. They actually bind very close to the phosphorylation site of the kinase, and by doing that, they block the release of pro-inflammatory cytokines. But because of their relative size, they actually almost fall over and block the phosphorylation site, such that the positive feedback that you see in inflammatory conditions is blocked. And how does that come? I mean, there must be classical MAP kinases. Oh, there's MAP kinase inhibitors have been into clinical development before and shown that they work. What's the difference? The difference is they were all small molecules, and small molecules bind at something called the ATP site of the kinase. And by binding there, there's an enormous amount of crosstalk. And what they showed previously with those MAP kinases was a waning of the effect over time, and also they showed liver changes. We will not go down that route because we won't get liver exposure because we're peptides. We'll be delivering them topically to start with, and I'll come on to how topically, but topically to start with. And we know that if there is any systemic exposure of these peptides, they will be broken down to their naturally occurring amino acids. In terms of amelioration of the small molecules, amelioration of the effect, the waning of the effect, that was because there's a positive feedback from the pro-inflammatory cytokines. They positively feedback onto the kinase and re-stimulate the inflammatory process. What we do is different. We actually block that positive feedback, block the phosphorylation of the kinase. So we shouldn't see the waning of the effect. And we, from a safety point of view, we're a peptide, and we predict that we won't see. I have to stop saying we won't because we haven't done it, but we predict that we won't see the safety side effects that we're seeing with other kinases, and of course with all the other anti-inflammatories. So you will have a study with a classical MAP kinase as a control, or as an arm. You'll have your peptides. What's the proof? What's a real good proof of concept experiment? The proof of concept in a clinical trial, or a proof of concept in the tissue biopsies? On the journey towards the clinical trial. The proof of concept we see is taking inflammatory disease tissue and in the lab test our peptides against a positive control, which will be a small molecule MAP kinase. They're easily found. A steroid, because steroids are most commonly used for inflammatory conditions, will have a negative control, and of course we'll have a non-treated control. It's a complicated experimental process, but actually it's actually quite simple if you break it down. At the end of the day, you'll have to show safety, so you're going to be looking at the therapeutic window, aren't you? You're going to be looking at the so when in the time frame are you going to get a handle on whether there is this greater safety margin than with a classical MAP kinase? The safety margin we believe we will achieve, because we deal with nanoparticle delivery. We're dealing with tiny doses compared to standard small molecules. the way that we'd like to do it is to demonstrate it works in human tissue first of all. We're on that road, and then we'll go into the sort of full preclinical development. So we're 2 to 3 years away from entering the clinic.
Great. And how much are you looking for, Mike?
So we've received this Innovate UK grant. We've received a little bit of seed funding. We're looking for about $500,000 to $1 million now. Of course, the more money we can get, the more we can do. But $500,000 now will add to what we've already received and allow us to look at 4 different tissues. We're going to look at Burns tissue, which is what we're looking at with the Innovate UK grant. We're going to look at dry eye disease as an option, which is a mouse model. We want to look at atopic dermatitis, which is a skin disease that is poorly managed at the moment with topical steroids. And we'd like to look at inflammatory bowel disease as well. That's what the initial, but then from that, we'll have data that will give us the lead indication for the lead drug.
But your critical question is one of a safety margin. That's really what you're looking for, an improved safety margin over existing MAP kinase.
Well, there are no MAP kinases on the market because the MAP kinases, the small molecules, all stopped in phase 2 of clinical development because of safety and because of the waning of the effect. Principally the safety, the liver changes that were seen. We won't see liver changes, will be topical. From a safety point of view, we'll be in smaller doses because we're nanoparticle delivered, and because we're peptides, those peptides, if there is systemic exposure, will be broken down into amino acids.
Fantastic. Now I've got Graham doing me a big tea, which makes it time for me to shut up. Mike, you're always a pleasure to talk to, my friend, and we're always
Thanks, Tony.
To see you. Let's ask questions to our three friends. Graham, you can curate them.
We'll go with Tom first.
Hi, it's lovely to hear your enthusiasm. I've listened to it for a number of times now, and it continues to get better, even though, you know, there's blue skies in Manchester, or light blue skies in Manchester. but you know, one of the things with regard to efficacy and nanoformulations is that the nanoformulations increase the bioequivalence of the compound. So do you think you may see systemic effects?
So do we think that we'll get systemic effects from delivering it topically, say topically to the skin, potentially topically to the lungs? The short answer is no, because if those peptides reach the systemic circulation, we know that the systemic proteases will instantly break those peptides down to amino acids. Of course, we'll have to prove it in preclinical development. Of course we will, but we do not believe that given topically, we will see a safety issue.
Interesting. Michelle.
Thanks,
Yeah,
Mike.
I actually had a little bit the same thoughts like Tom actually has, like, because you have a nanoparticle system which delivers the peptide, you have a protection of the peptide. So if we think about the indications you just mentioned, they all are disrupted skin or disrupted gut and tissue and lining. So there's clearly exposure to potential systemic compartments. So I'm not 100% sure that you won't have any systemic delivery, but clearly you'll have to demonstrate that indeed. I'm curious about like what your next step would be in terms of the development of this, because clearly inflammation, you call a couple of indications, but there's a lot more areas of inflammation. Are you thinking of specific targeted organ delivery, like for example, cardiac or neuroinflammation or anything like?
The way we're thinking at the moment is literally topical delivery. So topically to the skin, atopic dermatitis, burns, anti-scarring potentially, topically to the lungs by inhalation, asthma, COPD, to name a few, ventilated pneumonitis, which is quite in the news at the moment for obvious reasons. Topical to the gut by gel for rectal proctitis, or delayed, once we show that clinically, then delayed release capsule. You could deliver it topically to the eye for dry eye disease as eye drops, topically to the ear, potentially to help with age-related deafness, topically to the nose for rhinitis. There's
so no plan for systemic really yet. Okay.
Not at the moment. That's a possibility certainly later on, absolutely. And the benefit of potentially the nanoparticle that we're using, you can target with that nanoparticle.
Great. Michael, round off the session.
Hi, Mike. Good to see you again. Just building on sort of question from Tom, Michelle on the safety. I just wondered at a high level how things in terms of experiments gone in the de-risking of this approach, even though it's topical on the safety aspect.
I think it's a great question because safety is very important, particularly if we're going to be topical, particularly if we're going to go for skin diseases such as atopic dermatitis, and that's where our selling point will be on safety. Safety can be viewed from many different directions. It can be, of course, related to dose of drug. You can, which happens very commonly, issues with small molecules. We've shown cellular efficacy at 100 nanograms per mil. We believe that tissue efficacy will be 100-fold, maybe 1,000-fold greater than that, but we're still in 0.1 of a microgram. We're not ever going to, I think, be above a milligram, for example, and that's topically. From safety, a lot of the safety and the safety pharmacology and all those work will happen later once we've demonstrated, and this is the key bit, once we've demonstrated this works in human tissue via a topical approach. We'll literally take the biopsy, put the formulation on, and measure a reduction of cytokines released from the inflammatory tissue, from the inflamed tissue. Once we've shown it works topically in human tissue biopsies, we know it's going to work when we get to the clinic. Thereafter, we'll solve problems of formulation, problems of stability, problems that could happen in drug development. Of course they do, but we'll always know it's going to work when it gets to the clinic. Safety is important. Where low doses will be in micrograms per mil, I predict. We're peptides. We will systemically, there will not be systemic exposure. The nanoparticle that we are using at the moment is made from grass products, so we don't see that as a major issue.
I think we need
fantastic
to finish, and we need to thank Mike, because you're always a pleasure and a delight to talk to.
Thank you very much. Thank you.
A lot more. We will talk a lot more, and we're going to meet you again. But I need to wind it up, Graham, because I think the moment is.
We do.
Thank you so much for coming to the second version of our XY Factor. I think it was a fabulous event. I hope you all enjoyed it. Thank you to Tom. Thank you to Michelle. Thank you to Michael. Thank you to all of our speakers. Thanks to everyone, really. And keep coming. Tomorrow, what's that?
Well, we've got 6 o'clock. We got one in 50 minutes, Tony. So we're back on in 50 minutes.
Tomorrow.
And then tomorrow at 9, we do have this talking about people and executive search. And then we have a Tony section. We have a free section at 12 o'clock. And then we're going to be having Pelago at 3 and we're going to talk about the XY Factor winners at 6 o'clock. So you've got to come to that, and we're all very excited. So we're looking forward to the judges.
Any of the speakers go through Graham. We've got carrier pigeons, we have smoke signals, we have any way that you need to get in touch. And thanks again to everyone. And I really hate you, Graham, for saying that my skin isn't beautiful, because it clearly is. I'll see you folks. See you later.
Bye everyone. Thanks.
Thanks a lot.
Bye-bye.
Bye.
Bye-bye.
All the best.
See ya. Thanks, guys. Thank you very much.
Thank you. Bye.
Bye-bye.